Hi, my name is Setayesh Yazdani, and I'm a master's student at Structural Genomics Consortium at University of Toronto. Today, I'll be talking about mapping the genetic variants of SARS-CoV-2 onto its protein's crystal structures. Viral genomes are genetically unstable, which means that they can easily develop mutations that are associated with drug resistance. And it's important to look at SARS-CoV-2 sequences and map the mutations onto its protein's crystal structures. Due to possible emergence of MERS-like or SARS-like coronaviruses, for instance, from the bat strains that circulate in bat reservoir species, which could potentially transmit to humans and be the cause of future outbreaks, it's very important to do a broad survey of coronavirus proteins in order to identify the best strategies for viral inhibition. By mapping genetic variability onto crystal structures, we can inform the design of broad-spectrum inhibitors. First, we identify the mutations across thousands and thousands of SARS-CoV-2 samples. Then we map those mutations onto the binding sites of the proteins. And here you can see that a binding site that is heavily mutated should be avoided and not a good target for a broad-spectrum inhibitor. The second case, where we do not see mutations, which means that it's a more stable binding site, should be prioritized. And in the last case, inhibitors should be designed in such a way that they avoid chemical interactions with the site of mutation. So the goal of our project is to identify genetically stable binding pockets that are less likely to mutate and reveal the most promising chemical inhibition strategies. We so far looked at 3 targets of SARS-CoV-2, but in this talk, I'll be mainly focusing on the main protease. And if you would like to have a read of the details of the methodology for papain-like protease and NSP16, please refer to my posts on openlabnotebooks.org. Genomic RNA of SARS-CoV-2 is about 30 kilobase, and which is 2/3 of it is directly translated to polyproteins, and those polyproteins are then processed into functional pieces via the main protease and the papain-like protease. So that's why they're very important in viral life cycle, and they're important targets for anti-SARS-CoV-2 therapies. First, we identified the only druggable pocket of the main protease. Here shown in blue is the catalytic site of main protease. The D score, or the druggability score, is 0.88. That indicates that it's a difficult site on the druggability spectrum. But it's important to note that there is a catalytic cysteine residue in the main protease catalytic site, cysteine 145, that has been already exploited by covalent inhibitors. After finding the catalytic site, then we find the surrounding residues, and we do that by looking at side chains within the 2.8 angstrom vicinity of the catalytic pocket wall. Here we found 21 of them. Then in the next step, we have downloaded sequences from UniPro database, and we identified 27 unique sequences with different percentage sequence identity values to the main protease sequence. Then we made a diversity dendrogram while focusing on the main protease catalytic site and just looking at the selection block that we are interested in, which is the 21 residues. And here you can see that we got the consensus profile that is shown using colored symbols with varying sizes, which reflect residue conservation. So across the 21 residues, and we see that only 11 out of the 21 are conserved, meaning that 48% of the amino acids across this 21 are not conserved when we look at the 27 entries of beta coronavirus and alpha coronavirus entries. I want to highlight an important residue, and that's methionine 165, which is deep into the pocket, and it's buried by the inhibitor. Looking at just 7 entries of human coronaviruses, we can see that methionine 165 is not conserved. And genetic variability at methionine 165 is a potential liability for broad-spectrum inhibition of main protease, both across past and future coronaviruses. Our collaborator, Nicola Di Maio from European Bioinformatics Institute has identified genetic variants at SARS-CoV-2 main protease catalytic site. He identified very rare number of mutations, and based on our predictions, they're expected only to affect inhibitor binding very minimally. And we also have shown to you two positive controls, methionine 165 to isoleucine and glutamine 189 to serine. And so you can see that the corresponding change in Gibbs free energy and the effect that it has on inhibitor binding for the methionine 165 mutation is very significant. Basically, based on our precision method, any value greater than 2 kcal per mole is considered significant. The similar sequence of analysis was followed for the pan-like protease and also NSP16. And here I'm just highlighting some of the important findings. NSP16 was the most druggable in terms of its catalytic site. And also, it had the highest number of conserved residues at its catalytic site, followed by main protease, then papan-like protease. Looking at the SARS-CoV-2 variants at proteases' catalytic sites, they're only predicted to affect inhibitor binding slightly. We are going to continue our work by looking at more SARS-CoV-2 targets. Now, I would like to thank my supervisor, Dr. Mathieu Shapira, also our collaborator, Nicola Di Maio from European Bioinformatics Institute, and also we would like to thank all the authors who have kindly deposited and shared genome data on GSET. Thank you for your attention.